Chapter 3, Section 2 discusses the molecules of life, of which there are 4 main types. These molecules that are underlined here are the polymers or macromolecules, the large molecules. These items listed on the right side are their monomers, or their building blocks that will make these larger ones by undergoing condensation reactions. So the first one is carbohydrates. Carbohydrates are made up of monosaccharides, mono meaning one, and saccharide means sugar. So carbohydrates are made up of single sugars. Proteins are the second molecule, and they are made up of amino acids. Lipids are the third main molecule, and they are made up of fatty acids. And the last one are nucleic acids, and their building blocks are called nucleotides. So our bodies are made up of all 4 of these types of molecules. 3 of the 4 you can find in various foods that you eat. If you look at the nutrition label, the lipids are what you're going to find under the fats section. There's different types of lipids that you might find. The sugars are going to be in the carbohydrates section. And the amino acids are the building blocks of the protein. So we're going to see all of those available on most of the foods that we eat. The only ones that we're not going to see are going to be the nucleic acids. So our first macromolecule here, the carbohydrates, again, monosaccharide means simple sugar. That is the monomer or building block. One of the most common ones is glucose. A disaccharide then means that there are going to be 2 sugars that are attached to each other. So 2 monomers. An example could be sucrose, which is table sugar, like the normal sugar you would have at home to put in your iced tea or bake cookies with. And then polysaccharide, just like a polymer, is 3 or more monomers. This is going to be 3 or more sugars that are attached to one another. Some common polysaccharides are starches, cellulose, and glycogen. Now, starch is just glucose molecules that have been linked together. So like potatoes, you've probably heard them talking about as a starch. They have these glucose molecules that are all bonded together to make that molecule. Cellulose is also glucose linked together, and that's what we find in plants. It helps to give it some strength and rigidity. If you bite into a stick of celery, you get kind of that stringy, hard texture to it. That's partly due to the cellulose in that plant. And glycogen is also chains of glucose, and that's actually how animals store glucose. They will store it in this glycogen form in their liver and their muscles to be used later. Proteins are some of the most diverse molecules that we have. Within a protein, we are going to have amino acids. Those are the monomers. There are 20 different types of amino acids that can be arranged all differently to make us different proteins. The proteins make up, most of our skin and muscles. They can act as catalysts in plants and animals. And the way the amino acids are held together are through these peptide bonds. That's what's going to hold them together and give them their structure. And similar to the disaccharides and the polysaccharides of the carbohydrates, if we have a dipeptide, then that means that we've got 2 amino acids bonded together. And if we have a polypeptide, that means that we've got 3 or more amino acids bonded together. So another name for a protein then would be a polypeptide. That's just a chain of amino acids that are all attached to one another. This is just a diagram of an amino acid being bonded with peptide bonds to each other to form a protein. So this big long chain is the actual protein. Each of these little circles is representing the amino acid, and they have one kind of blown up so you can see the group. You've got the central carbon, and then there's that NH2 functional group that I mentioned earlier in this chapter. Proteins also have the ability to act as enzymes. Enzymes are molecules that will help to speed up reactions. They can be called catalysts, which a lot of the processes that happen in our bodies wouldn't happen fast enough just naturally on their own. They need a little boost from these enzymes. So they help by reducing the activation energy, or how much energy is necessary for a reaction to happen. They'll reduce that energy and allow these processes to happen more quickly. You can generally tell something is an enzyme by its ending, if it has this ASE ending to its name. So protease. As soon as you see that ASE, that should give you a signal that that is probably going to be an enzyme. So the enzyme protease helps to digest proteins. The enzyme helicase helps to break down our double helix, or our DNA. And enzymes also have the ability to change if the temperature, or pH, changes. So it's called a lock and key model. You can see here a little bit better in this diagram that an enzyme is designed to fit certain molecules. Just like, your key is supposed to only fit in your lock at your house. You hope that your key doesn't open your neighbor's house, or your neighbor's key open your house. Okay, you want it specific. And that's how enzymes work too. They have specific molecules that they will help to catalyze. Well, if the temperature, or pH, changes drastically within that organism, or their homeostasis is not maintained, then that can alter the shape of this enzyme. And if that shape changes, then it will no longer fit the molecules that you need it to react. So that lock and key model will no longer work. So that's another reason why it's important for us to maintain our homeostasis, so that these enzymes can keep functioning properly for us. pH, you've probably talked about before in science classes. Basically, you just need to have a general idea that pH goes from 1 to 14, and it's going to tell us how acidic or basic something is. Acids are going to be below 7. Bases, or alkaline substances, are going to be above 7. And then things like water are going to be neutral, which would be right at 7. Our third main molecule of life are lipids, and lipids are made up of fatty acids. Those are the monomers, and fatty acids are made up of a couple different parts. They have a hydrophilic end and a hydrophobic end. Now, if you think of phobia, you think of something that's scary. Well, that's exactly what this is. Hydrophobic means water fearing. So this tail end of this structure, this fatty acid, is nonpolar, okay, similar to oil. So it does not like water. It does not dissolve in water. They also have a hydrophilic, or water-loving, head structure to them. So this portion is polar, which could dissolve in water, and the tail, or these little leg structures, are nonpolar. And lipids, actually, believe it or not, are going to be the molecule out of these four that we're discussing. They are going to be able to store the most amount of energy for us, even more so than carbohydrates. There are some more complex lipids. We mentioned these fatty acids here. These fatty acids can be arranged in what's called the phospholipid bilayer. That's what helps to protect our cells. This is the cell membrane. Here's that hydrophilic head, here are those tails, and when they're arranged in twos, that's called the bilayer. And that's going to allow some substances to come in and out of our cells, and also provide us some protection about things that we don't want to have come in and out. There are also things called waxes. Okay, you've probably heard of earwax before. They provide a kind of a protective covering. So what you might think of necessarily as gross, some wax is good. It helps to protect our inner ear from microorganisms. Different types of fats are complex lipids. They're called triglycerides, which are just chains of molecules. You've probably seen saturated and unsaturated fats on food label. The saturated ones are the ones you want to avoid the most, and the unsaturated ones are not terrific for you, but still a little bit better than the saturated fats. And another one of the complex lipids are steroids. Steroids such as cholesterol or testosterone, which is a form of a hormone, those are also different types of complex lipids. And finally, we've got nucleic acids. Nucleic acids are built of nucleotides. That is their monomer, and that's what this structure is right here, a nucleotide. It has a sugar that is the central molecule here. Phosphate group is attached to it, and a nitrogen-containing base. Now, there are two different types of nucleic acids, DNA and RNA. In DNA, the sugar is going to be deoxyribose, and in RNA, the sugar is going to be ribose. And then there are varying nitrogen bases that could be attached here to help provide some variety for our nucleotides. And this is a picture of the DNA structure. It's that double helix that you might have heard of before. The main function for DNA is to store essential cell information, and it contains all of our genetic information and codes. And RNA is mainly to help to store and transfer information required for making proteins.